And so I was supposed to give a sort of a research talk, but I enjoyed the tutorial talks yesterday so much that I'm going to, so I spent a bit of last night to actually change the talk to make it more like a hybrid tutorial and research talk. So the first part will be more tutorial, and I'll talk about some of the more recent stuff that we're doing in my group in the second half. So feel free to ask questions during the talk. So I'll be talking about some of the applications and challenges of deep learning in genomics. So many of you have heard about the various quite impressive achievements recently of deep learning in a lot of different applications. Right, so I mean, some of the earlier applications from a few years ago was in image recognition. And then more recently, there's also a lot of interesting advances in natural language processing. Right? So a lot of the speech recognition or image-to-speech captioning or speech-to-speech translations. Under the scene, there's a lot of these different deep neural networks. Right? And there's also like more ambitious kind of plans going forward. Right? So there's all these activities, research activities, going about trying to develop self-driving cars, right? So that involves different aspects of really robust image recognition systems, robust sort of reinforcement learning systems, right? And behind the scenes of that, there's also a lot of these deep neural networks. And many of you also probably heard about these, AI systems that can play games at very high levels, right? So it was made a lot of headways in the last year or so, where researchers from DeepMind developed sort of these games based on reinforcement learning and deep reinforcement learning to play Go, right, and also to play other games like Atari games. And they achieved a really impressive performance, surpassing currently the best human players. Okay. So when you think about these kind of applications of deep learning, there are basically two broad classes of kinds of applications. Right, so the first class is what we're more familiar with, sort of making predictions. So that's also what we call sort of supervised learning in machine learning. Right, so making predictions, is just really what you think of when you think about machine learning. Right, so you have some input examples. Right, so in this case, it could be images, or sort of natural images. In other examples we'll look at, it could be sort of genomic sequences or medical images. Right. And the predictions usually have sort of a few well-specified categories of classes or labels that you want to predict for each image, which class does it belong to. Right. So for this image, what's the probability that it's a particular kind of car? Right. So that's sort of the supervised learning approach. And this sort of makes up, I think, sort of the bulk of machine learning. Right. So there's actually this other aspect of deep learning, which is in more recent years also becoming quite important. It's more the unsupervised learning approach. So this is the second category, where the goal here is not to make predictions. Right? We're not trying to classify an image or classify a genomic sequence, but we're just trying to actually model the whole distribution in a way that allows us to generate new images or new DNA sequences. Right? So here's a cute example of this, where as input, right, you just have a human that sort of draws out the outline of different kinds of handbags. Right, so these are cartoon outlines that the human draw. Right, so you put this as the input into the algorithms. So these are the ground truth. That's the middle column. And what the algorithm tries to do is it learns the whole distribution of these different handbags, right? And then it's able to, just from the input, basically try to draw a sample, right? Just for a conditional sample from the input. So the right column are the samples that it draws based on the cartoon sketches. And you can see that it actually, the sample it draws are actually very realistic, but it also has sort of interesting variations from the actual, the original true images. Right? So these are sort of the more unsupervised learning examples. So I'll talk a little bit about both classes of applications of neural networks. So in the last few years, so it's clear that there are a lot of interesting ideas in these deep neural networks. And people have started to explore the applications of them in various biomedical data. Right, so if you think about applications, maybe the most natural places to look for applications would be sort of more in biomedical imaging. Because there you're also already dealing with images, and so it's easier to think how these algorithms can just be ported over. And people have had a lot of quite also impressive achievements in biomedical imaging. So this is from also some students and colleagues at Stanford, where they're looking at applications in digital pathology. Right? So the idea here is that you're given some slides, right, of different kinds of tumors or different tissue samples. So currently, in the hospitals, it's a relatively manual process, and it's a labor-intensive process for the doctor to individually look at the slides and try to classify these slides as to whether they're pathogenic or whether they are benign or malignant. Right. So here this research group basically had obtained a very large data set of these labeled examples. They're able to train an algorithm that has basically is able to surpass the human pathologists in terms of the accuracy. Right. And so related kinds of approaches can also be applied to other applications. For example, here, this is from Andrew Ng's group, where they're trying to take EEG signals and try to detect abnormal patterns, right, arrhythmia. So in my group, we've also been doing recent works along these lines. So one of the big problems in building cardiovascular models is you actually want to have very good three-dimensional shapes of the different vessels, right? And currently, that's also very much a manual process where you have the humans basically has to go in and essentially do a lot of the segmentations and annotations by hand, which just takes many days to build one model. So with some colleagues at Stanford and students, we are able to use variations of these models, basically develop systems that can also automatically generate these three-dimensional models and segmentations of different vessels. Okay, so these are sort of in these imaging applications. As we said, somehow it's easier to think about why the methods that are developed for image recognition can be applied to biomedical images. But what we really want to talk about today is how do these methods actually apply to genomic data, right? Which seems to be quite different in characteristics and different properties. So would these methods even work? So the talk will have two parts. The first part will be basically a tutorial of sort of basically how do these neural networks work. I'll go through a little bit more detail of a particular class of neural networks called convolutional neural networks. And then the second part of the talk, I'll try to illustrate some of the main lessons and messages that we learned from working with these neural networks in genomic applications. Right, so there's a lot of sort of the hype about these different neural networks, about deep learning. I think it's really important when thinking about applying them to new settings to think about what's really the real content of these methods versus what is sort of just the talks and the hype. Right? So it's still very much a work in progress, but I want to sort of get across maybe sort of three messages or lessons that we learned from our experience. Right? And I'll illustrate each of these messages with sort of a concrete example based on our recent research. So the first message is that when thinking about applying them to genomic examples, it's really about the training data, more so than the actual models and the algorithms, right? How to build the proper training data. So the second message, and I'll go through each of this in much more detail. The second message is that somehow being able to make predictions is good, but it's not really the most exciting applications, right? So these generative models, being able to sample, is actually, I'll try to convince you, it's actually much more exciting than simply being able to make predictions. Even though most people are working on predictive models, the generative models, I think, are the more exciting future directions. And the third message, maybe it's a little bit more provocative, is that everybody is thinking about sort of overfitting with respect to these neural networks, right? So you have big networks with millions, hundreds of millions of parameters, right? So overfitting should be a very big problem. So what I want to convey here is that actually overfitting is not the main problem, right? So it's basically a solved problem for these neural networks. But there is the main bottleneck, which is what we call the fragility of these networks. And I'll define precisely what fragility means. Okay, so here's a relatively quick introduction to these neural networks, just so that we're on the same page. Right, so the basic unit of this neural network is an artificial neuron. Right, so we'll start from the very beginning. So we have a neuron, just think of that as taking a weighted linear combination of your inputs. Right, so the input into your artificial neuron is just a vector of real numbers. So the neuron would have weights assigned to each of the inputs, right? And it's basically doing a very simple weighted linear combination, a weighted sum of these input examples. right? And then taking a weighted sum, and then it's going to basically pass it through some thresholding function, right? So in this case, it's going to basically set everything that's below 0. If the input for the weighted sum is less than 0, it's going to set it to 0, right? So it's going to be just simple threshold, and that's the output of 1 unit. Right, so a unit of this neural network, artificial neuron, is extremely simple, right? It just takes the input, weighted average, threshold, and then that's the output. So the whole idea of these deeper neural networks is basically taking modular compositions of a lot of these individual neurons into different architectures. So a particular popular kind of architecture for working with imaging and for genomic data is basically what are called convolutional architectures. So I'll explain what that means. Right, so suppose that I have an image that I want to classify. Right, so the image is, let's say, it's pixels, right? So 32 by 32 matrix, and there are also 3 colors, RGB colors. So it's 32 by 32 by 3, so a 3-dimensional matrix. Right, so that's my input. Right, so each entry of the matrix corresponds to the pixel value at that position. Okay. So what a convolution neural network is trying to do is basically defining a lot of these local convolutional filters. Right? So think of each filter as being a little patch of a patch of those artificial neurons that we saw from the previous slide. Right? So here I have this 5 by 5 filter. Right? So there are 5 by 5 of these artificial neurons that we saw before. Right, and what we're going to do is that we're basically going to scan each of these patches across the image. Right? For every place where I put this patch, it's going to take this weighted linear combination of the pixel values at that position. And it's going to spit out a number. That's the output. Okay. So the operation is very simple. Right? So I'm just going to scan this filter, right, made of artificial neurons across my image. Throughout the image. So that's the convolution operation. Right, so the output of this is going to be sort of a smaller matrix, 28 by 28 matrix. Right, it's a little bit smaller because some of the edges are left out when I do the convolution. Right. So each filter now is basically just a set of these artificial neurons. And when I scan across the image, it's giving us this output, which is basically another image. Right, you can think of this as another matrix or another image. So I don't have just one filter, so I can define a lot of different filters. So each filter is going to have a different set of weights, so it's going to be taking a different linear combination of the input values. Right? And when I scan a different filter, it's going to give me a different 28 by 28 output. Right? So if I have a lot of different filters, then I'm going to get a bunch of different outputs. Right? One corresponds to each of my filters. And this is where the modularity becomes very important, right? Because now if I just look at the stack of outputs after taking this convolution, it's basically just another 3-dimensional matrix. Right? So I can think of that as basically another 3-dimensional image, and then define apply additional convolutions on top of that image. But this convolutional layer is basically the basic operation, the unit of operation. Is that clear? Are there questions about that? Okay. So there are basically 2 essential operations when building this neural network. So one is the convolution operation that we saw on the previous slide. The second operation is basically sort of a downsampling. People give it pooling, which is sort of a fancy name for it, just taking averages or downsampling. Right? So what that means is that if you have sort of a relatively large image, I'll just do the simplest thing, which is I'll sort of downsample it into sort of a smaller image. Okay, so here's basically an example of how you actually apply this and how this would work on real natural images. Right, so this is my input here. Right, so it's a picture. But you can think of that as a 3-dimensional matrix of pixel values. Right, so here I defined a convolutional network with a few different layers. Right, so each layer basically corresponds to modularly taking a convolution operation, exactly as we saw before, and then taking sort of using some pooling operation or doing sort of downsampling and averaging. So it's actually quite interesting and it's a lot of fun to look at what's the outputs of each of the layers. right? So in the beginning, you can see when you take the convolutions, you're basically capturing some of the edges, right? If you take the convolution, you take this thresholding, you're finding some of the edges of the image of the cars, right? So it's basically, each of the convolution is basically a little edge detector trying to find the local patterns. And as you go later on, right, so then the patterns actually becomes harder to interpret, right? So some of the later layers are essentially taking sort of combinations of these local edges, right? So that's becoming a little bit harder to interpret. But at the very end, it is giving something that's easy to parse, right? So it's basically doing sort of a logistic regression at the very end. It's taking whatever feature that's learned, and it's basically doing sort of essentially as a logistic regression to say, what is the probability that this whole image, it's a car, it's a truck, an airplane, right? It's assigning a probability to each of these classes. Right? And we train this network, right? You basically go backwards this process, right? So you have examples where you know the actual labels, and you essentially do sort of a gradient descent, right, to train the weights of the network, right? So the parameters of the network are essentially the weights assigned to each of these neurons. Okay. So that's a very sort of brief overview sort of what are these, how do these neural networks work, right? So conceptually, it's easy. I think I find it helpful to just keep a few sort of bullet points in mind, right? So there are a lot of, when you read these papers, there are a lot of different architectures that people think about, a lot of different training algorithms. But in the end, just think of these neural networks as being sort of a very flexible functional approximation, right? So you have some input, you have some output, and when you try to build predictions, either from whether for imaging data or for genomic data, you're basically trying to learn a mapping, right? Some function from the input to your output. So a neural network is nothing but a nicely parameterized, flexible class of functions to do that, to approximate to your true mapping. Okay. So the network, there's no magic here, right? So you can't just put in garbage and then try to get out some nice results, right? So in other kinds of machine learning, or in Bayesian methods, right, you have to put in your prior information into the structure of the graphical model or into the priors. Right? So here you still have to put in your domain knowledge, your priors, essentially into how you build the architectures of the network. Right? So in that sense, it's not fundamentally different from other sort of statistical machine learning models. You have to put in your priors, domain knowledge into the architecture. Right? So in particular here, right, so where did we put in the prior knowledge? We put in the prior knowledge in that we think that there are local edges that should be somewhat sort of spatially invariant. Right? That's why these convolutional filters, convolutional operations, that particular architecture is appropriate for this setting. Okay. And if you use the wrong architecture, so that'll be analogous to if you put in the wrong priors, and then your models can actually be very much worse off. So probably the biggest advantage of using these kind of neural networks is that it is a very well-engineered large-scale platform to do large-scale optimization. Right? So there are a lot of other machine learning techniques that have sort of the first two bullet points that allows for flexible functional approximations, that allows you to build into priors. But for various reasons, right, so these neural networks are actually just have been extremely well-engineered, partly because they're used in a lot of these large companies. So as academics, then we get to basically leverage the results of what are this very well-engineered system. So that's one of the main benefits. So a caveat here is that actually to train these models, it typically, not always, but typically requires actually a large number of well-labeled training examples. Okay, so that was for images. I showed an example for images because it's sort of easier to interpret and easier to sort of see what's going on. But what we're really interested in today is how do you actually apply these methods to genomic data? Right, so, and for this, I think it's also maybe it's helpful to keep in mind sort of this analogy. Right, so the image that we're thinking of, right, is the input. So in the genomic context, we want to think of the DNA sequence as basically being the images. You can think of the DNA as essentially a very long, one-dimensional image. Right. So when we saw that for images, right, basically what these algorithms are looking for are local edges, local filters. So in genomic context, that's analogous to looking for local subsequences, right? Local structures, or more, that's what we call motifs. And the output, right, the labels of these images, rather than being cats and dogs or airplanes, right? So what's relevant for us here would be whether this particular stretch of DNA sequence, right, whether it's bound by by a transcription factor, whether it's maybe relevant for a particular disease, right? So these are the outputs. So okay, so just to make this sort of analogy from this here like very clear, very concrete. So here's a particular, the simple illustration of how you would actually apply this for DNA sequence data, right? I'm taking basically the same kind of architecture that we saw from the previous slide, right? These convolution architecture that's applied to images. And we're going to see how this applies to a DNA sequence. Right, so as we said, right, you think of the DNA sequence as basically being an input or a one-dimensional image. Right, so there's a particular way, so in the images, right, you have 3 colors, RGB. In the DNA sequence, where every nucleotide, we have 4 different base pairs. So it's also analogous. So it's encoded basically as this sort of as this very long matrix. Exactly as before, right, so we have these local convolutional filters. So each filter, again, is going to scan across the image, looking for particular substructures. Right, so the nice thing about these filters is that they are spatially invariant, right? So they're going to be the same filters applied to everywhere. So in this example, each color here corresponds to a different filter, right? So different filters have different sets of weights. So they're looking for different subsequences, essentially looking for different motifs. And then the output of the filter are basically just going to be, some after doing thresholding, it's going to be some sort of quantification count of at that particular location. Did that filter detect this particular motif or not? Right. So that's going to be the output. And then there's maybe some additional linear combinations of those outputs. And you can iterate this. And then in the end, the prediction I want to make is, what's the probability that for this particular sequence, whether there's a protein or transcription factor that's binding to this sequence. Right. So conceptually, the model is very similar to what we saw before with the images. And you can say that, okay, so this is actually not all that different from what people have been doing, right? Which is true, right? So, people in computational biology and genomics have been working with these different motif scanners, right? Which are essentially what these convolutional filters are, right? So I think it is very similar. So the main difference here is that by allowing for these additional compositions, of convolutions and additional layers, it's basically giving us a little bit more flexibility in trying to learn not just different motifs, but maybe different combinations, interactions between different motifs. Right, this is where having this flexible architecture, modular architecture, becomes quite useful. Okay, so the output, right? So once we have this set up, then it's you can go to town with this, right? So somehow the output doesn't have to be whether just predicting whether it's bound by a protein or not. You might want to also predict what's maybe the expression of some target genes or of nearby genes, right? Okay, so here's why I want to maybe go through in more detail sort of these 3 messages about these neural networks that I want to convey, right? So the first message is that, especially when dealing with genomic data, it's really much more about building the right training data rather than a specific model and algorithms. So we looked at this model before, and let's say if the goal is actually to predict whether the sequence is bound by a protein or not. Right? So here it's actually we have a lot of data sets, right? From from large-scale ChIP-seq studies. So the input here is typically on the order of 1,000 DNA, sort of a vector of 1,000 DNA. Nucleotide, so 1 kb DNA sequence. The output is whether this particular sequence, particular maybe candidate regulatory region, is bound by a TF or not. And here, what's nice is that you can, so we have a lot of ChIP-seq data across not just many cell lines, but also across many TFs. Right, so that's basically can be used as sort of the gold standard labels, or at least for the algorithm, sort of labels for what is being bound and what is not being bound. Right, so typically you get on the orders of millions of positive examples of actually binding events from these biological ChIP-seq studies. And for the negative training sets, right, so negative events where things are not bound, so people usually take genomic sequences that are not overlapping different histone modification sites or different ChIP-seq binding sites, right, as negative controls. And when you look at this training data set, then so it is actually a very large training data set, right? And it has a very clear label, right? Whether oftentimes it's binary, whether it's bound or not bound. And it's actually roughly on the same order of magnitude as the data that's used to train these large image data sets, right? So here you actually have enough training data, well-labeled training data to make some progress. And I should mention here that in a lot of in our experience and in a lot of our colleagues' experience, right? So, and the actual specific, exactly how you build the architecture ends up not being so important, right? So it is important to build in the priors that maybe you're using these convolutional structures and you're setting the filters to be the right length, maybe about 10 or 12 base pairs. But beyond that, it really doesn't matter so much in terms of the performance or predictive accuracy of the algorithms, whether you use 3 layers or 5 layers, or exactly how you train the model, right? So this is where it is much more important to have a well-curated training data set where it's clear what are the positive and negative examples and what are the labels. And once you have that, then actually most of the standard architectures will work relatively well. And it doesn't need to be very deep architectures. Okay, so I want to go through maybe a more concrete application example of this, something that our group in collaboration with Anshul have been working on recently. Right, so here, just to illustrate the ideas we saw from the previous slide. Right, so here the setup is that I have some reporter assay, right? So I have some synthetic sequence, maybe I take from different parts of the genome, or I can just generate, right? Maybe I want to test those sequences. And we can hook those up into different reporters. Right? These are just genes where we can have readouts of the gene expression when you put this whole construct into cells. And what's nice about this is that you can do this very much in parallel, right? So you can get in one set of experiments on the orders of 100,000 of these labeled examples, right? Where each labeled example is one set of this construct, or one set of DNA sequence, paired with an output, which is the expression level or the activity level of the reporter gene. And what is nice here is that you can test a lot of different DNA sequences with different combinations of reporter genes. Right? And you can also test this in different cell types. Okay, now let's translate this into our example, right? So the input here is basically this sequence. Here it's shorter than 1,000 kb. 1 kb is on the orders of maybe a couple hundred base pairs. Right? So you have this sequence that's the input into the algorithm. So the output of the algorithm, the target for prediction is basically the activity level of this reporter assay. Right? And we have 100,000 or a few 100,000 of training examples. So it's quite good training data. And basically, the setup then is fairly straightforward conceptually, right? Basically, we're just scanning this convolutional neural network, similar to the one I showed you before, across this construct. And every position in the construct, it's going to try to predict what is the activity score of this gene when paired with this regulatory element. And the nice thing about this is that once you train this model, so you can actually get quite high prediction accuracies around validation or test data. Once you train the model, you can actually use it to make interesting interpretations. Right, so here's one kind of interpretation. Right, so now I have trained this neural network, so the weights are fixed. Right, so now it is just a deterministic function from input to output. Right, so I can now apply this neural network in a particular position, right, or a particular regulatory element. And it's going to predict some maybe like a low activity level, low expression value. And then computationally, we can do this sort of in silico mutagenesis very efficiently. Just say, okay, so I can take this a particular nucleotide of interest, maybe change it to a C rather than a T, and it's feed that the mutated sequence back into the prediction algorithm to see, okay, so what is this? Does its predicted activity actually change or not? So maybe if I make a particular mutation just to this one nucleotide, keeping everybody else the same, and, maybe it gives a sort of a high prediction score, predicted activity score. So the thing about this, which is nice, is now I can do this very systematically, right, across all of the regulatory sequences, right? So there are computationally efficient ways to essentially do these different in silico mutagenesis. And what that allows you to do is to sort of a simple way to interpret what is the thinking essentially behind these neural networks, right? So it's essentially assigning an importance score to each nucleotide based on how much the predicted activity level will change if you mutate in silico that particular nucleotide. And that actually does have quite strong agreements. Is there a question? Yeah.
So a quick question. What would be a good way of generating a significance value for that? Like some sort of key value?
Yes, that's a good question. So what one can do is to actually have sort of control sequences, right? Sort of background sequences where, so I talked a little bit about these negative sets, essentially where they don't have any predicted, or they don't have any measured activity scores. And you can say control, you can see, okay, so if you just make random mutations there, the assumption being that most of those mutations are unlikely to actually lead to a generation of sort of new enhancers, just by sort of point-wise mutations. And then you see, okay, so does that actually, you have false positives there. But what I'll actually mention is that there are other ways you can sort of assess the statistical significance of these importance scores. All right, so one way is to say, so here the x-axis corresponds to the importance scores of the nucleotide. Right? Now you can apply this network to the whole genome, right? So I can just apply it to the genome, to the human genome, and assess the importance score of the nucleotide. So that's the x-axis, and the y-axis is basically the conservation of that nucleotide. Right? So this is applying this model to sequences it's never seen before during its training process. And it is doing fairly reasonable things, right? We can see that base pairs that have both very, very positive or very negative, meaning that they're predicted to either be highly influential in up or down regulating the expression, also have much greater sort of conservation scores. So another way you can sort of use to sort of interpret the results of this, right, is to see, okay, so how do these different importance scores correspond to different epigenetic signals, chromatin marks? And the idea here is that so there are specific chromatin marks, without going into too much detail, that corresponds to sort of highly, where if you have a high signal at those marks, then it's more likely that you have different regulatory activities. Right, so what we can do here is to basically bin the true genomic sequences by their observed chromatin signals from the highest peaks to the lowest peaks. And what you see here is that so basically the peaks that have basically the highest predicted importance scores also have the highest sort of DNA signal. And so there's a very clear monotonic relation between the importance scores predicted by the algorithm with the actually measured chromatin signals. Okay, so that was hopefully now you've seen some examples of how you can try to apply these models to make predictions, right? If you have a reasonable training data set, then this gives you a nice machinery to try to both make predictions and also by doing this in silico mutagenesis to interpret the outcomes of these networks of the predictions. So the second message I want to try to discuss is that making these predictions is nice, but somehow it's not really the most exciting, I think, applications of these methods. And in many ways, these predictive models are they're conceptually fairly standard models that we all know how to do pretty well. Okay. But I want to go back to these examples of the generative models, which I think are actually sort of more exciting and potentially the more powerful applications of these neural networks. Right, so again, in this generative example, what makes it quite striking is that you just have to give it some sketch of what the kinds of things you're looking for, right? And the network is actually some rough specifications. The network has learned the distribution of these objects, of these handbags, well enough that it's actually able to generate synthetic samples that actually fits your prescriptions, but also fill in a lot of the details. Right, so why might this be exciting? So imagine now if we actually want to design particular proteins or DNA sequences that have particular specifications. Maybe it wants to bind well to certain proteins and very specifically. Right, so those are the inputs I want to put in, and I would like the network to basically automatically generate sequences that satisfies those specifications and generate a lot of diverse set of them. Right. So currently, it's not clear how to do this sort of by hand in a principal way. So I want to discuss a little bit, give you some intuitions for how these generative models work. I guess these are actually much more recent advances in the community, and I think they're quite exciting. So the idea of the generative model is that I would like to basically design a network, a neural network, that can capture and can model a very complicated high-dimensional distribution. Right, so suppose my distribution is a set of images. Right, so that's a very complicated distribution in a high-dimensional space, and I would like to have a neural network that models that whole distribution. Here I'm not trying to make predictions, I'm just trying to model the distribution. So the idea actually turns out to be very simple. So let's start with a distribution that we're all very comfortable with. Let's start with maybe a high-dimensional Gaussian distribution. And I can sample from this Gaussian distribution very easily. So what we like to do is to basically learn a deterministic mapping, a function, call this f, from my Gaussians to this really complicated high-dimensional space, right? Such that the density of the Gaussians, after I do this mapping, would actually capture the densities of my images or of my DNA sequences. Right, so that's the idea. So we know that these networks are good functional approximators. Right, so let's just parameterize this function f by sort of one of these convolutional neural networks. But there's still a bottleneck here, right? So I mean, the whole difficulty of this is that if I'm trying to model the space of sequences or space of images, I don't actually have any explicit form to know what is the distribution, right? And that's the whole crux of the problem. So how do I even know that I'm transforming my Gaussians in a way that's actually reasonable, giving a reasonable probability of distribution? So the very nice idea here is let's actually try to learn that. So here's how it would work, right? So suppose I have a sample from my Gaussian distribution at some point here. Let's say we fix my mapping, right? I'm going to apply this point, just pass it through my mapping, and it's going to give me maybe some sample. Right? In this case, it gives me a pretty good sample, and it actually looks like a real digit. And if I have a different point, right, maybe it gives me a different sample. So the idea here is that, okay, so I want to be able to tell you whether this sample I get from my distribution after I pass it to this mapping is actually a realistic sample or not, right? If it is a realistic sample, that means that somehow my mapping, this transformation f, has captured a lot of the interesting densities and distributions of my set of images. If it's not, then it hasn't done well enough. Right, so how do I do that? So why don't we just actually have another classifier whose goal is to basically try to predict whether the synthetic sample that's generated by this algorithm, right, is actually a real sample or a fake sample. And this classifier is given access to a set of other real examples, real images. Right, so the classifier is trying to predict whether it's real or not. Right, in this case, maybe the classifier says, okay, so the image generated by the algorithms actually looks pretty realistic, so it couldn't tell the difference. Then this particular parameter value is somehow rewarded. Right, basically given a little bit gradient boost. Maybe in other settings it generates sort of a bad example that's clearly not sort of a human drawn image. Then the algorithm, the classifier, is going to predict this to be fake. and then this, this parameter value is going to be punished, right? So it's going to go down in the gradient. Right. So again, this is sort of conceptually very simple, but it gives you sort of a nice modular way to, by iterating through this process, to see whether to basically learn a setting of the parameters so that your original Gaussian distribution is mapped to something that closely approximates your full distribution of images. Right? And once you have that, then to sample, it's extremely easy. I just sample from a Gaussian, pass it through my learned deterministic function f, and then I have my image or I have my protein. Okay, so recently we've been exploring how to apply this to actually two genomic synthetic biology or synthetic genomic examples. Right, so the examples that are along the lines that I mentioned before. Right? So let's say I want to design DNA sequences, and I would like to have some specifications. Right? Maybe someone comes in, maybe one of my synthetic biology colleagues comes in and says, okay, we want to have DNA sequence that binds specifically to these proteins X and does not bind to Y. Okay, so I should mention that this whole system is called generative adversarial networks. And adversarial because you can think of this as being sort of a game between the generator, who's trying to generate images, and the classifier that's trying to say that whether the generated images is real or fake. Right? There's a competition between these two objects. Okay, so here, right, so the ideal outcome is that, right, so this network is going to take these specifications and then basically sample from the conditional distribution and give us this really nice diverse set of DNA sequences that satisfy these specifications. If you could do this, then this would be really powerful for a lot of biotech applications. Okay, so basically the strategy that we use is similar to what we saw before. Right? So we have a generator that's essentially taking Gaussians and then try to map those Gaussians, try to work them in some way to approximate the more complex probability distribution of DNA sequences. And then we have a discriminator or a classifier whose goal is basically try to reward or punish the examples by telling it whether it's real or synthetic. And here the goal is to basically, like a simple goal, for example, would be try to see, okay, can we generate proteins that actually look realistic? Right? This is a proof of concept. Right? So in this case, then the discriminator is basically trying to generate DNA sequences that codes what it thinks are basically realistic proteins. And then the discriminator is trying to classify those proteins as being, well, does it look real, or does it look fake compared to the existing database of human proteins? So what, it's still actually, there's a lot of work to be done in this area, but we've shown that it's, as a proof of concept, that you can actually go to the system and generate proteins that look very realistic. Right? So there are a lot of different ways you can measure sort of how realistic the protein is. So one simple way is basically in terms of its foldability. Right? So if you have a protein, does it actually fold into sort of stable structures? Right? So that's a sort of a fairly simple but computationally efficient way to evaluate how good this protein is. And in this work, we're able to show basically you have this network that can synthetically automatically generate artificial proteins, and it's actually have sort of foldability scores that essentially look as good as the foldabilities of the real human proteins. Right? It's actually having sort of even more higher foldability scores, so a little bit more stable structures than the distribution of the naturally observed human proteins. So I should give a shout out actually to my students, Anvita Gupta, who's an undergraduate at Stanford, and she actually won the national sort of invention competition, collegiate invention competition for building this generative model. Okay, so the last part of the talk, the last message is that I want to convince you that this sort of prevalent thinking that overfitting is sort of one of the main bottlenecks with these neural networks, especially with these deep neural networks. But I want to show you that actually overfitting is really not the main problem, right? We know essentially how to deal with overfitting, but actually there's a much more complex problem called the fragility of these networks. I mean, they're actually intrinsically very fragile objects. And this is also very much work in progress. I think there are lots of interesting ideas research ideas here, potentially for graduate students. So, so basically, we know how to prevent these neural networks from overfitting, right? So the standard definition of overfitting is that sort of how, like, the test accuracy. Well, so we know they're not overfitting because the test accuracy is essentially the same as the training accuracy. Right? When you have very clean test holdout data. Right? So if I give an example it has never seen before from my test data, it's going to predict that this is sort of a panda with fairly high confidence. Right? So it's not overfitting by the standard definition. But it's extremely fragile in the following sense, right? So if we give it sort of a very small perturbation, it's not a random perturbation, but I can tell you how the perturbation is constructed afterward. But if you just give a very small perturbation, then the same network is going to predict this image to be, given, which is the type of ape, with almost 99% confidence. Right? Whereas to humanize, there's clearly nothing really has changed in image. Sure. A few more minutes? Okay. So the idea here is that these networks are essentially causing a lot of, well, so why does this happen? Right? So we think these networks are actually causing a lot of deformation to the space. So here's an example actually made from our colleague, David Duvenant, where if we take, let's say, so imagine if our input space is just this two-dimensional grid. Right? So you pass it through different compositions of functions, but as you increase the number of layers or number of compositions you have these horrible distortions of your input space. Right? Why does this matter? Because if you take 2 points that are close to each other in the original input space, right, the image of the panda and something that's very close to it, they're going to map to places that are actually could be very far apart in their output space. Right? And this is sort of also an example. This shows like how the distortion changes as the depth of this algorithm, as depth of the function increases. But also we see that the distortion is actually increased during training time, right? So you train this algorithm. This is sort of nice work from my student, Amir Atta Gorbani, where this is again the two-dimensional input, where there's a few different classes, right? This is sort of the distribution that I want to model. And basically, what he's showing here is as you train the network, here's a two-dimensional output of this after you pass it through the network. As you train the network, right, so the network is actually learning over time to really deform the space, deform the input space in more and more complex ways. Right? Again, it's going to cause a lot of distortion in that points that are nearby in the beginning are going to be sent to very far away places in the end. And this is a huge problem, I think. This fragility is a huge problem for a lot of the applications of this approach, because as you can imagine in image settings, right, if I'm trying to build these classifiers to predict whether an image from a pathology scan is malign or healthy, right? So if a very small perturbation of that actually changes my predictions with high confidence, then that's clearly problematic. Right? And similarly for the genomic prediction examples. So there's a lot of things here that I'm not going to go into, but I think this is a very interesting and important open area of research. And if any of the students here are interested, I'm happy to chat more about that. Okay, so here are my students who did most of the work in these examples. Thanks.